Microfilaments, Intermediate Filaments, and Microtubules by Franco and Sehi. Microfilaments, the what and the where. Microfilaments are also known as actin filaments because of the material that composes them. They are very long and thin by cellular standards, usually about 8 nanometers in diameter and ranging up to a few micrometers in length. That may not seem like much, but is actually several thousand times how thick they are. Microfilaments are located just beneath the cell membrane, giving the cell strength and some rigidity. Microfilaments. Function. The main function of microfilaments is to provide strength to the cell, but it also anchors the centromeres during mitosis and can even interact with myosin to create motion in the entire cell. The microfilaments also link some proteins from the surface of the cell to further inside of the cytoplasm. Microfilaments and their interaction with other parts of the cell. During mitosis, microfilaments anchor the centromeres, causing stability and allowing the cell to split. Microfilaments also interact with the proteins by allowing them to connect to others elsewhere in the cell. The cell would be very fragile without microfilaments, and some, such as white blood cells and certain amoeba, would be unable to move without the help of microfilaments. Microfilaments. Interesting fact. Microfilaments can move through a process known as treadmilling, which is progressively losing actin monomers on one side and gaining them on another. Intermediate filaments. Intermediate filaments are named so because they are intermediately sized, in between microfilaments and microtubules. The purpose of intermediate filaments is to provide support throughout the cell, especially to hold the nucleus in a net of safe fibers. Intermediate filaments. Interesting fact. The intermediate filaments compose up to 85% of the dry weight of certain cells.
Microtubules are straight, hollow cylinders that are made out of alternating layers of alpha and beta tubulin in a helical structure. Microtubules are used in locomotion because they make up the cilia and flagella of cells. They're used in mitosis and meiosis because they form spindle fibers to separate the chromosomes. And all materials used inside the cell are transported along the microtubules. Microtubules can be found almost anywhere inside or outside the cell. Microtubules often interact with other organelles to perform their job. For example, the mitochondria provides ATP to power the cilia and flagella. Microtubules are an integral part of the cell. If they stopped working or did not exist, the cell could not perform any of its normal functions. Microtubules, microfilaments, and intermediate filaments are grouped together because they all provide support and structure for the cell. Here are some interesting facts about microtubules that you might not have known. They can change length by polymerization or depolymerization, and they can grow up to 1,000 times longer than they are wide.